introduce everyone to our next speaker, Dr. Susana of Collaborative Drug Discovery, based in the UK. Dr. Susana holds a PhD in computational chemistry from the University of Warwick, where she gained experience in molecular simulation. Her skills in molecular modeling were developed further by pursuing 2 postdocs at the University of Southampton and the University of Cambridge, UK. Susana's career in the software industry started with ligand-based drug design and QSAR analysis. Susana is now the Senior Application Scientist at Collaborative Drug Discovery, also known as CDD, providing training and technical support for CDD Vault users in industry and academia, and working drug discovery projects throughout the CDD customer base. So, ladies and gentlemen, you really want to take into account this presentation because their platform can really help accelerate your scientific study. And with that said, the floor is yours.
Thank you, Sam, and hi everyone. Thank you again for the introduction. So yes, I'm a senior application scientist at Collaborative Drug Discovery, and I provide technical support for CDD customers all over the world. And as mentioned earlier, my background is in computational chemistry, and I've been with CDD for over 3 years now. So today I'm going to talk about, I'll start my presentation mentioning some of the current challenges that scientists face nowadays in drug discovery. So these challenges are in terms of infrastructure and data, too many systems, tools, and lots of data. We are faced with the issue of having multiple platforms. Science is much more collaborative nowadays, which means that quite often we have collaborators on the other side of the world. So how can we exchange data with them in a secure and easy way? Other challenges might include the cost and maintenance of resources, and in addition, having multiple tools brings usability challenges in terms of difficulty onboarding and the need of power users. So in terms of data challenge, we have the example of the drug discovery process. This overall process includes a timeline of multiple steps, and this is an example of Lilly's website. So various types and large number of entities that have to be cataloged, hundreds to thousands of different kinds of assays that are run on these entities. And this often results in millions and millions of individual data points that have to be organized and mined. And this large amount of data has to be differentially accessed by many scientists, some of whom for business or security or IP reasons should only have a limited view of this information. In addition, numbers of documents, notebooks, reports, and summaries that, again, with their own security and access needs. So basically, the challenge is dealing with a mix of different entities, molecules, biological entities, proteins, plasmids, DNA, antibodies, multiple types of data, raw data, high content screening data, and to add to this complexity, a variety of tools. Managing data across multiple systems and tools is hard and time-consuming. And CDD Vault has over 17 years of experience in storing, managing, and organizing chemical and biological data in a collaborative environment. It's a flexible platform which you can use to store your molecules, your proteins, DNA, and other biological entities along with the assay data. It can be seen as your own single source of truth, your ecosystem where all your internal or external collaborators can access through an easy-to-use web interface. So CDD Vault combines the features of multiple data management systems into one easy-to-use cloud-based platform. It is intuitive to use, it requires no IT support, and training is provided at no cost. With CDD Vault, scientists can organize their multiple types of data and entities through an intuitive GUI. And there is also the ability to connect your favorite third-party applications with a RESTful API. So you get the get, put, and post API calls, which enables CDD Vault to be the center of any ecosystem of software tools and applications. So the modules included in CDD Vault are the activity and registration. This allows users to register entities and assay data. And historically, CDD is best known for managing small molecule data. And even though that is still our strong point, it is now possible to register biological entities like cell lines, plasmids, and antibodies. We have an inventory module to keep track of the debits and current amounts of your batch compounds, and a visualization tool to plot, filter, and mine the data. Also, an ELN to register your experimental procedures in a flexible way. Okay, so in summary, CDD Vault is intuitive, secure, and collaborative. You have full control of what data is private and what is shared with the collaborators using our CDD Vault unique project-based collaboration. So with that, let's take a look. So I'm now just going to share my screen one more time, and we are moving into CDD Vault. So this is our web platform. So this is our website, collaborativedrug.com. And there's no need to install any software in your computer. All you need is your internet connection and modern browser. Once we log in using email address and a password, we reach this landing page. So currently I'm logged in my private secure vault in the same way as thousands of users log in every day in their private secure vaults. We can see a list of projects on the left-hand side. So this is a unique feature in CDD Vault, which encourages and facilitates collaboration across scientists. So across your internal or external collaborators, you can share just the data that you want with in each project. So each project can have different members, and the members will only view the data of the project that they are part of. I'm going to show you an example of the types of data that we can store in our private secure vaults. So this is a report table. It has 3 molecules. We can see the substructure highlighted, which was used in my query. And we can also see a molecule ID or molecule name, which is automatically generated when a structure is registered in the database. We have a section here with the properties. So the chemical properties are calculated, up to 20 chemical properties are calculated when a structure is registered in the database. I'm only displaying the molecular weight, but if I want to, I can click in Customize your report, and then I can add the other chemical properties to this table. These properties and all the registration is done with Chemaxon, which is our chemical cartridge. In the next section, we have the molecule and batch fields. So these fields are created and customized by the Vault administrator. The Vault administrator has full ability to decide which fields they need in their vault, and then the users, they can populate these fields when they register their molecules in the vault. Also, so these fields, they can be number, text, they can be a file, attachment, they can be a date, but CDD Vault will also generate automatically links to the ELN. Whenever a specific molecule or batch is referenced in the electronic lab notebook, CDD Vault will automatically generate these links. So in here, we know that batch number 1 of this compound was used or was referenced in this particular ELN entry, and I can follow the link I already have it open in here, and then I can see the full experiment. So this is actually a synthesis where we have the analytical data and the chemical reaction, and of course, because the compound was referenced, we have the link in the report table. We also have the inventory, which keeps the current amount for the batch. And in the next section, we have the assay data. So all the assay data that we have, that we obtain from our experiments, this can either be in vivo, in vitro, or in silico experiments. They can be stored in a protocol. So the concept of a protocol is, in essence, a container to store the assay data. Plus, we have the ability to, or CDD Vault has the ability to perform several analyses, such as dose response plots, calculate IC50s, and then perform calculations on the readouts. You can also use the concept of a protocol simply to store single point screening assay data, and then you can perform averages or other types of calculations on these readouts. So this is an example of the types of data that we can store in CDD Vault. In this example here, this is a vault which I am using to store my small molecules. But as mentioned earlier, CDD Vault can also be used to store biological entities or any other types of entities. So if you have plasmids or antibodies or anti-drug conjugates, as we have in here, you can have a separate vault. Then when we log in, we can simply switch between vaults in this arrow in here. So we can switch between vaults, we can also search across vaults, and we can link the entities between vaults as we have in this example in here. So in this table, we have 5 antibody drug conjugates. The reason why we suggest or we recommend to have separate vaults is because the business rules for these antibody drug conjugates will be different from the business rules from a small molecule. And therefore, you can have multiple vaults in your own ecosystem where you can register your entities with the different types of entities and business rules. In this report table, we have the entity name, we can see the synonym, and different batch fields and entity fields, along with the assay data performed on this particular ADC. What is useful and interesting is that one of the functionalities in CDD Vault is that we can link our entity into entities in other vaults. So here, for example, I have this antibody drug conjugate linked into a small molecule in an oligo vault, and also we have the payload also linked in a separate vault. So basically, I can simply open this tab and I can view the information for this particular payload from my ADC. This is now in my payload ADC vault. I can view the structure, all the molecule, the batch fields, and also all the molecule fields. So this is just to say that you can use your CDD Vault to store your small molecules and biological entities. In the interest of time, I will now move into the visualization, which has been updated recently, and I thought it would be nice to show you. So this is a report table with several molecules and many columns of numerical data, chemical properties, and assay data. So we can click in Launch Visualization, and a new tab opens up. We have a 2D scatter plot with 4 parameters, and from here I can simply change my x-axis as you would. We can change the y-axis in here to any property that you may like. We have this settings wheel on the top left where I can assign a color to a different parameter. We can choose the range of colors, and we can also bin the color. So in this case, bin the molecular weight. I can define here how I want to see. We can also add more bins, and we can also assign a shape to a different parameter. So as you can see here, I have now all my legend is in here. All these data points are chemically aware. So if I hover over the dot, we can see the structure. If I want to pin this structure in the plot, I can just do shift and then, shift click, and then I can simply kind of keep the report card of this molecule in this plot. We can also customize the properties that we see in here, again in settings, tooltip, and I can maybe just select other properties that I want to view in my card view. There is also a statistics tab where I can just add a linear regression in here to my data points. We have the filters on the right-hand side. I can add other filters from this table, and then I can just kind of filter my data points from here. We can add multiple plots in the same page. I may want to add another scatter plot. I may want to add an histogram as well, where I can go into settings and basically change the binning, add a parameter as we wish. And we can have up to 4 plots in the same page. All these plots are automatically, they interact with each other, well, in the sense that if I just select a small portion of this histogram, all the other ones are being automatically updated. And then we can always save these high quality images into PNG or PDF, or we can also save these as Excel. You can also launch a session. So you can import a file, either a CSV or SD file, and then you can just filter and mine your data. Okay, so this is kind of a small overview of the Vault. I'm now going to stop sharing one more time, if I find the button, and I'm going to share again, and we are going back to the slides. So let me just share again. There we go. So what I would like to show you, talk about now, is just to talk about of a case study, and that basically I will talk about COVID Moonshot, which has been using CDD Vault. And COVID Moonshot is a global consortium that aims to rapidly develop antivirals for COVID-19. And it's a partnership of more than a dozen organizations around the world, which have united their efforts on the race towards the discovery of new therapeutics to tackle this pandemic. It has been advertised as an ambitious mission to produce a COVID antiviral, and therefore the name COVID Moonshot. As we all know by now, COVID-19 is caused by a virus, SARS-CoV-2, and this picture is a representation of when the virus infects the human host. RNA is translated into this long polyprotein, which needs to be chopped into the different viral proteins. And in SARS-CoV-2, there are 2 proteins which do this cleavage. We have the PL-Pro and M-Pro. And in the COVID Moonshot, the focus has been on the M-Pro protein, which is the main SARS-CoV-2 protease. So things actually have moved pretty fast since last year. The structure of M-Pro was solved. And then Diamond was also able to solve a new structure of the SARS-CoV-2 main protease M-Pro, and performed a large fragment screen against it, which resulted in over 70 fragment-bound structures. All results were made public straight away, so scientists could use this data as soon as possible. And basically, these results highlighted the idea that somewhere in the merge base there must be a compound which is an inhibitor or antiviral. So the actual project COVID Moonshot involved a crowdsource of ideas from other scientists. So through an online platform called Postira, the medchems could submit a molecule or a fragment that they believed that would bind to the SARS-CoV-2 protein. And this workflow involves 3 steps: design the molecules, the synthesis of the molecules by enamine, and the test of the molecules. And this is where CDD Vault has helping to manage this process. There are basically 3 projects in the COVID Moonshot Vault. So as we saw earlier in the demo, the idea, we have this concept of projects within CDD Vault. So basically there were 3 projects, a virtual project to capture all the design molecule ideas, a synthesis project for all the molecules that are decided to be synthesized, and the made project for all the compounds that are made, basically. Of course, that things are usually not that simple as it may look, and there are many other steps involved in the process, which basically involves checking for duplicates, checking whether the synthesis is feasible, perform in silico analysis such as docking and scoring, and prioritization of the synthesis of compounds by enamine. Then the compounds made are tested in different assays, and all the data from the various assays has been stored in CDD Vault. Also, the raw data for the dose response calculations are imported into CDD Vault, and the curves are also performed with CDD Vault. Then the data is being pulled from CDD using the API to bring the data back into the public forum. So plate data, as we can see in this image, has been imported into CDD Vault, and those response plots were performed within the platform. And also all the ELN the ELN has been used to capture all the procedure of the experiment. Last year, there was this paper. This paper came out, and just last week, COVID Moonshot received an 8 million grant from the UK's Wellcome Trust on behalf of their COVID-19 therapeutics accelerator. So this has been obviously a very important project. Okay, so I'll finish here. So these are some examples of our customers which have been using CDD Vault, both large-scale collaborative projects, but also small organizations and academic groups. So I thank you for your attention, and if you have any questions, I'll be happy to answer.
Thank you so much, Susana, for your insightful presentation. And we do have a question from Mr. Dmitry Pavelk from researcher from Salon Pharma in Poland. He's asking, is it possible to do bulk upload for molecules from a database?
Absolutely, yes. So I did not show on my demo, but we do have them. We can do bulk import of molecules either via the GUI using the import data tab or via the API. Absolutely.
Wonderful, wonderful. And if anybody has any other questions or comments, you can type it here. Also, we can connect you with Susana and her team via email to schedule the demo and learn more about their platform. As she's shown, they have a lot of collaboration within the industry, and their software really can help you accelerate your scientific research goals. So I'll give everyone another moment if they have a question. Okay, yeah. And if any other questions come after this program, Susana, I'll be sure to share them with you and your team. And again, on behalf of Precision Global, thank you so much for presenting.
Thank you so much. Bye-bye.
Bye.